Dr. Ray here, and I would like to spend a few minutes talking about the development of the forebrain. So in class and other movies that you might have watched, we talked about development of different aspects of the brain stem. And so we have the mesencephalon, the metencephalon, and the myelencephalon pictured here, and we spent a lot of time devoted to discussing how they have unique appearances and cross-section and the different characteristics that are in each section. But now we're going to talk a little bit about the forebrain. So here in this light pink region is the portion of this developing neural tube in this figure that will develop into the diencephalon, and then the bright pink reddish color are the vesicles that are growing laterally outwards from the most rostral aspect of this neural tube, and this is going to become the telencephalon. And so there'll be two sides to it, and you'll write a right hemisphere and a left hemisphere eventually. And I like this kind of cartoon drawing because it's simple, and it's good sometimes to bring it back to the big picture to understand. Remember that the cavity inside the tube is going to develop into the ventricular system in each of these areas, and it's going to look very different. And the reason why the shape of the brain ends up being so distinctive in different places is because of the amount of growth of neurons and axons in each place, and the differential growth of that one area compared to the areas that have grown around it. And so the cavities inside these telencephalic vesicles are going to become what we call the lateral ventricles. And so that's why you end up having two lateral ventricles, and then you have one third ventricle, which has some contribution from the telencephalon, but most of it will be involving the diencephalon, and then you're going to have one of the cerebral aqueduct and one fourth ventricle. But because of these vesicles growing outward, you end up having two lateral vesicles. And I like this figure here. You can see, let's see if I can change my pointer here. I like this figure a lot for appreciation. Here you have the midbrain, and you have that midbrain flexure here, which curves, as you can see, the telencephalon growing and very large and kind of pointing more ventrally. And then you can see this pontine flexure, which is in the other direction, and then the cervical flexure here at the medulla. And so I kind of like this view, and it's also early enough view that the cerebral hemispheres coming from the telencephalon, they haven't really overgrown the rest of the brainstem yet, and so you can really see everything as its distinctive part. And so the forebrain is going to have two major divisions. One is the telencephalon, and the other one is the diencephalon. And so you'll be learning all of the different parts of the telencephalon, and essentially there'll be like regions of the telencephalon. And then here you can see in this early view of a developing diencephalon that it has three main parts that start off being rather big bulges, but in the end, really the middle one, the thalamus, is going to sort of maintain a larger size compared to the smaller epithalamus and hypothalamus, although they start off all three about the same size. So you can see the area of the diencephalon in this cut view. You can see a laterally placed telencephalon growing outward in its open communication in the ventricular system with the rest of the brainstem here. So here's another view of the diencephalon. So this is a sagittal cut, and you can see why we call epithalamus above, hypothalamus below, and thalamus here in the middle. Because of its orientation, it looks a little bit different in the fully developed brain, but it's named really for its orientation here at this point. And this is a frontal or coronal section, and you can see how these swellings of neurons in the wall of the diencephalon are going to develop into the the thalami and the epithalamus, you can see how thin the roof becomes and how large the third ventricle remains. And so this thalamus that is going to maintain the biggest part of the diencephalon is going to grow inward in the diencephalon, and in 70% of the cases there'll be an interthalamic adhesion between the two parts. So not everyone is going to have one, but 70% of the time they'll be adhered to each other. So here you can see the location of the thalamus, which you'll become more and more comfortable with the more you study the brain, looking at a cadaveric images and also looking at stained sections. And this is a sagittal section. I just wanted to start you off with basic. Where is the thalamus? The thalamus is located here in this location. So this is the brainstem, and then as the brainstem comes up, it is going to flow into the region of the diencephalon, and then essentially the telencephalon grew outward from that aspect. This would be the most retial aspect of the neural tube, the lamina terminalis, and then the telencephalic vesicles grew outward from one side to another. And then the green region I have highlighted here is the hypothalamus. You'll be learning a lot about its function and the nuclei that are situated in the hypothalamus, and then you can see the mammillary bodies. This is one of the characteristics of this region that is positioned here, and the part of the optic chiasm that's sticking off here from the hypothalamus. And then this is a region of the epithalamus here. You can see it's much smaller than what it originally started off being, about the same size as the thalamic swelling and also the hypothalamus. Hypothalamus is sort of medium size, and this is the region of the epithalamus. And you'll learn a little bit about structures around there like the pineal gland later on as well. So the pineal gland is just going to be one median diverticulum in the diencephalon, and it's just good. The cells are going to proliferate into a solid gland. I know you already learned about this in histology, but if you've kind of forgotten about its appearance, you might want to look up its histologic appearance, and you'll talk a little bit about its function later on in this course, but it is part of the diencephalon. And I like these pictures here from the textbook because you can really see in this frontal or coronal section, this is the location of the thalamus, just to give you some reference. And here you can see the telencephalon growing out on each side. This is a choroid plexus, which is going to be a special type of epithelium that will make cerebrospinal fluid, and we'll talk about that in another course, another section. And you can see the cavity of the telencephalon is very large at the beginning, and it has a very open, wide communication with the third ventricle to start off with early on, called the interventricular foramen. It's going to narrow down and constrict, but you can still see the interventricular foramen in lab, so you will be able to look down into the lateral ventricle and see where it's going to communicate into the third ventricle and drain the cerebral spinal fluid that is being produced. And so there is another portion of the telencephalon, on the floor of the telencephalon, of gray matter called the corpus striatum. And you'll be learning a lot about these nuclei and gray matter. But for development purposes, what you want to know is that this area is going to be proliferating neurons a lot. And I'll get to that slide, I guess, in a minute. And the rest of the telencephalon is more free to grow in size and is going to be growing bigger and faster. And so this area, although it's proliferating, it is not going to be widening and getting larger in size as much. And so it's kind of, the telencephalon is kind of constricted at this point, which causes the rest of the telencephalon to grow faster and actually maintain more of a C shape in the end. And that will become apparent later on. I have some better pictures to show you more of that. And so here you can see at first there's wide communication with the third ventricle from the lateral ventricles, and that they're going to eventually constrict down as this lateral ventricle and entire telencephalon maintains more of a C shape. The interventricular foramen is going to constrict down and become a smaller opening. And so the cerebral hemispheres are going to markedly increase in size, and the cranium is a limited cavity. And so what happens is you get a lot of folding of the cerebrum, and that allows for increase of surface area of the brain without having to increase in that much size. So at the beginning, the brain is less cephalic, which means smooth, and then it's going to become more and more folded throughout progressive weeks of development until you see gyri and sulci that are very prominent in the near birth time for the fetus. And then you're going to learn some of the prominent gyrus that are prevalent throughout this course and throughout lab. I wouldn't be too concerned about it right away. It can be kind of overwhelming, but basically just learn sort of the most prominent sulcus in the brain, which gyri you really need to know, and then you can add to your knowledge as you become more interested in certain regions for function too. So as the telencephalon grow progressively larger, they actually trap a little bit of mesenchyme between the growing vesicles, and that mesenchyme is going to become the Falx cerebri. So the dura mater that is going to be between the cerebral hemispheres. So just sort of little interesting aspect. You might wonder how this tissue becomes located where it's located at, and you might remember from Gross that this strong dura mater is going to fix the brain and kind of hold it in place, and as it comes forward and attaches itself to the crista galli. And so here, I showed you earlier in the picture because it was an actual histology slide, but here you can see in the figure the corpus striatum, the location of these structures, and which you will learn so mainly consists of the caudate and the lentiform nucleus, which is going to have the putamen and globus pallidus in there. And then in between the two, you have a projection of fibers called the internal capsule. And all of these structures are going to become sort of stretched as the brain grows and remodeled into a C shape. And so you might wonder why the shape is so unusual on sections, and it is unusual, but it's because of the fact that the rest of this telencephalon can grow freely, and this portion here is proliferating more but not growing as much in size, and it is more constricted in that region. And so here you can see this in the figure as we start smaller, and then as we elongate the telencephalon, that the caudate, which is this aspect here, is going to become longer and C-shaped, and so this is the situation in the position of the putamen. And with that, the ventricles are also going to become C-shaped because they just lie next to the corpus striatum. And so when you're learning about this, you might wonder how we ended up getting a temporal horn and temporal lobe of the telencephalon, but that's because of this curvature. And so this is just a summary slide that shows you pointing out all of these parts that are kind of just basic parts to get started if you're just getting started with the brain, because you start to look at the brain as a whole, but you can't see the inside. But as you look at the inside, you're looking at cross-sectional anatomy, and it's good to always have the 3D understanding of what the brain looks like in your mind. And so especially when you're starting off learning the brain, it's good to kind of do what's called interleaving. If you have the big picture is that the brain inside the telencephalon, you have these other structures, this corpus striatum and also the ventricles, and this is the general shape of it. Then you can start looking at cross-sectional anatomy and figure out where you're located at according to the characteristics of these structures here. And so this is just also the summary slide explaining what I mentioned about the internal capsule also becoming C-shaped. So I pulled out this image from the embryo project, the virtual embryo library that they have at LSU. And here you can see choroid plexus, you can see the Diencephalon, back here is the brainstem, and this is a region of the Telencephalon, the telencephalic vesicles, and I wanted to show you an up-close picture of the cortex. And so in this image, what you can see is that if you look at the cortex at day 40, you can see the neuroepithelium is prominent and proliferating, but not very organized. And then if you just look 16 days later, you can see these zones of neurons that are present. And so I think what I want to impress upon you is that the gray matter is going to be on the outside of the telencephalon when you look at the cortex. And the axons of these neurons are going to grow inward and add bulk to the brain. So the white matter is going to be on the inside. And if you look on the outside of the cortex, which we're looking at now, the axons have not been developed. What you need to remember is the opposite of the spinal cord. And also that damage can actually come really easily to this cortex, because if you have any pressure applied from the outside, it's going to actually damage neurons instead of damaging axons. And so that's sort of very different from the situation that we have in the spinal cord, and it's something that you might want to take note of now, because you can have a lot of different kinds of damage and injuries that are going to put pressure on the brain, and that is going to cause death to the neurons in that cortical region. All right, so in this picture, I just wanted to show you how the brain becomes more tortuous, and with this gyri here and sulcus, sulcus is going to be the depression, and the gyri is going to be the ridge. And at week 14, completely smooth brain, and by week 38, almost at birth or at birth, the brain has a lot of gyri. All right, and because of the growth of the telencephalon, it grows so fast, and the corpus striatum, which is in this region, it's deep, is actually not growing very fast. So here you can see the cortex is overlying the corpus striatum, and this is the area that's not growing in size as much. The telencephalon actually overlaps this area, and so we call this cortical region the insula. So it's kind of hidden from the outside. You can't really see it from the outside. You have to sort of peel the temporal lobe down and peel the parietal lobe a little bit upwards to be able to peek in on the insula. But that's where it's located at, and that's why it becomes hidden in the brain. Okay, so that's all I have for the Telencephalon and Diencephalon. I have other movies if you want to watch after this. Thank you.